Hello and welcome to remote learning for microbiology. So a couple things. Number one, I accidentally, well, not accidentally, but I left your DNA tests at school. So there's no guarantee when we're going to be back. The minute we return to the building, I will have those graded, but for the meantime, I am not going to be able to grade them until I have them. Number two, we are diving into our microbiology unit. We're starting with bacteria. So for this lesson, we're going to talk about the different types of bacteria, their shapes, and their structures. Um, so let's get started. So a couple of things to keep in mind. We want to think about what makes bacteria so unique. They are unlike anything else in the world. What makes bacteria a bacteria? How is bacteria simple and streamlined? So it's what makes it simple and streamlined? Why does it have to be that way? We often use bacteria as a model organism in the biotechnology industry. That's why we learn about bacteria. And actually, in a couple months, we are going to be using bacteria in a lab in biotech class. So we want to look at why it's used as a model organism. And then we also want to look at what features bacteria have that make them so good at evolving. They've been around the longest. They were here first. They're the, single-celled organisms. They were probably the first living things to evolve, and they're still here. So what makes them so evolutionarily successful? So first off, bacteria are prokaryotic, meaning they do not have any membrane-bound organelles. They don't have a nucleus, they don't have mitochondria, they don't have endoplasmic reticulum, none of that. They have DNA, they have cytoplasm, they have ribosomes, and then they have some kind of protective outer covering. Okay? So a couple other differences. Prokaryotes are smaller. They don't have a nucleus of any kind, as well as any other membrane-bound organelles. The flagella is structurally different. They might have an outer covering on top of the cell wall called the glycocalyx. They do have a cell wall, which usually contains something called peptidoglycan. They do have a plasma membrane. All living things have to have a plasma membrane as well as cytoplasm, but they don't have cytoskeletons. The ribosomes are smaller. All living things need ribosomes. All living things need proteins, so they need the ribosomes to make the proteins. Their chromosomes are singular and circular. They only have one circular chromosome. They reproduce by binary fission and they don't use meiosis. They reproduce asexually. There's no sexual reproduction. However, they do get away with a little bit of genetic diversity in the form of conjugation. So we'll take a look at a lot of this today. So again, basic bacteria structure. This is like the generic picture you're always going to see. DNA, ribosomes, cytoplasm, cell membrane, cell wall, and then often this outer covering, an extra outer covering on top of even the cell membrane and the cell wall. And then, of course, you also see a flagella a lot of the time. Flagella are for movement, so if the bacteria needs to move, they're going to have flagella. So first, we're going to look at the cell wall. There are two different types of bacteria. We'll talk about this more a little bit next time, but there are gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria have more peptidoglycan. Peptidoglycan is a very rigid carbohydrate, so it provides a lot of protection to the bacteria once it enters its host. We have to remember that bacteria, a lot of them, they want to infect. They want to get into a host and then multiply. So having this thick layer of peptidoglycan allows them to survive much longer. Gram-negative bacteria do have peptidoglycan, they just have less. And then, of course, they both have a plasma membrane. So this peptidoglycan makes up the cell wall outside of the plasma membrane. Now, most antibiotics attack peptidoglycan production. Why is this an effective way to kill bacteria? If we go back and look, If bacteria are not able to make this peptidoglycan layer, then they have no protection once they enter their host. So it makes it a lot easier for the immune system to attack them. Okay? And interesting note, so gram-negative bacteria are often the ones that are resistant to antibiotics, and that is because they have less peptidoglycan to begin with. So the antibiotics are supposed to be attacking the production of this, but gram-negative bacteria are like, ha, I don't really need it anyway. It's not as much of an effect as it would be on gram-positive bacteria. So again, some bacteria also have an even further outer covering. It depends on the function they need. So, for example, if they need to prevent against desiccation or drying out, if they need to stay hydrated, they'll have a capsule. If they need to attach to something, they'll often have a slime layer. But regardless, if they have it, it's going to provide a certain function to them and it's going to be the outermost layer. DNA on the inside, some of their internal anatomy, they have DNA, a single circular chromosome, bundled up into a region called the nucleoid. Again, it's not a compartment, it's just a region where the DNA is. Bacteria also can have these things called plasmids. This is also circular DNA, but it is outside of the bacterial chromosome. So in the chromosome, they have the genes they need to survive. Plasmids are bonus genes. Not all bacteria have plasmids, but if they do, they provide some kind of extra, kind of like superpower. Like they might give genes for antibiotic resistance, or they might give genes for just some other cool feature, producing a certain enzyme that they wouldn't have normally. So this is what they need to survive. This is what makes them that bacteria. And then the plasmids, if they have them, um, it gives them some kind of extra ability. Okay. So bacterial morphology, what we just looked at was more the anatomy. Now we're going to look at the morphology, kind of the identifiable characteristics, um, external characteristics. So bacteria have different shapes. The most common are the coccus, the bacillus, and the spirochete. The coccus is the circle, the bacillus is the rod, and the spirochete is spiral shaped. Now you can also add a little bit more clarification to the name. So staphylococci are a bundle of circles. Streptococci is a single file line of circles. Staphylobacillus would be a bundle of rods. Streptobacilli is a single file line of rods. So these extra words kind of add more identification. The other ones we don't really need to be familiar with, but we want to know coccus, bacillus, spirochete, staphylo, and strepto. Okay. So you can also have different types of flagellar arrangement. Remember, flagella provide movement. So if some bacteria need to move faster or differently than others, then they're going to have certain flagella. So you have your monotrichus, your lophotrichus, amphitrichus, peritrichus, and amphilophotrichus. That's a fun one. For example, E. coli are peritrichus. They have flagella all over. Naming bacteria. So bacteria get two names just like every other species on Earth. They get their genus and their species. So theoretically, there are many different types of Escherichia, but only one type of Escherichia coli. We can often abbreviate this to just E. coli. It's pretty obvious that this means Escherichia coli, but you can't do this for all bacteria. Right? There's Moraxella bovis, Mycoplasma bovis, and I think what's the bottom one? Mycobacterium? Whatever it is. And bovis is not specific enough. The name is always italicized. All species names are italicized. Homo sapiens, Escherichia coli, it gets written in italics. And now we can actually get even more specific. So species is not as specific as it gets anymore. Now we have strains. So E. coli keeps evolving, and now we have these different strains. This one right here, the 0157 hyphen or colon H7, this is the bad one. This is the one right here, that winds up in our chipotle and our lettuce and makes us sick. All of the other ones are actually supposed to be in our bodies. E. coli naturally lives in your gut and digests your food for you, but if you have this one in your gut, then it makes you sick. So we get to the naming of bacteria. Some names have to do with the shape: Staphylococcus aureus, Streptococcus pyogenes. This second name often has to do with the disease it causes, like Streptococcus pneumoniae would cause pneumonia. Vibrio cholerae, so Vibrio is comma-shaped, cholerae tells you it's going to give you cholera. Sometimes, however, the name has nothing to do with the shape. For example, you would think that this one would have spirochete, something in the name, but it doesn't. So the naming of bacteria was kind of wacky for a while, and it was never really standardized. So they all have these different names that we just kind of have to get used to. Um, a lot of the times it does have to do with the shape, a lot of the times it has to do with the disease, and sometimes it's just named after the guy who discovered it. Not only do we look at the morphology of the bacterial cell itself, but you can actually look at the morphology of the colony. So we grow bacteria on agar plates. Agar is basically a nutrient dense food for the bacteria, and we plate them on this agar and watch them grow. They grow differently depending on the type of bacteria. So, for example, I think this one is E. coli. Um, this one grows red on agar. This one grows yellow on agar. This one grows kind of clumpy and yellow on agar. So you can use the bacterial morphology to actually identify the bacteria. Let's say you go and swab a door handle and then you plate it on the bacteria, or I'm sorry, you plate it on agar. If it turns out to be red like this, then you know it's only a certain handful of, bacterial types. It's not going to be E. coli if it grows red. So it's a way to identify unknown bacteria. Look at the colonies. So colony originated from one single bacteria cell that then divided, millions of times to make this clump. So within this colony, there are millions of genetically identical bacteria. And then not only do you actually look at, the color and shape and whatever of the colony, there are also different characteristics of the colony. We do not need to worry about all these different names. I just wanted to show you how many different types of colonies can form as well. You can look at it from the side. You can look at what form it takes. Does it, make larger separate dots? Does it kind of spread out into branch-like structures? You can look at the edge. What does the edge look like? And this can also tell you what type of bacteria you have. If it forms something like this, then perhaps that's very unique and it tells you, it's only this certain group of bacteria that do this. So you can actually give a name to an unknown bacteria. Sorry, I have a problem with putting in funny pictures that I think are funny. Bacteria reproduction, they reproduce by binary fission. So this is asexual reproduction. Um, the cell grows a little bit. Well, actually, first the DNA has to be replicated. The cell grows a little bit and then it splits in half. It's pretty self-explanatory, right? You had that original cell, it has to replicate its DNA, and then it's able to split in half, and you get two identical copies of the original DNA. Some DNA can actually do this every 20 minutes. So their growth is exponential. And remember, this takes 20 minutes, and then these two will start to go through the same thing. So after just 40 minutes, you now have 4 bacteria because each of these then goes on to divide. So it's an exponential growth. If you start with even just one bacteria, within 24 hours, you're going to have millions, if not billions.